Hi, I'm Alexa Fredston. I'm a postdoc at Rutgers University in the research group of Malin Pinsky. Today we're talking about realized thermal niche tracking of range limits of North American marine species. I'm sure you all know that species are on the move. Species are shifting their ranges and tracking the climate conditions they historically occupied as a result of human-caused global warming. We have a lot of evidence for shifts in the center of a species range and community turnover that are attributable to local changes in temperature. But what managers and stakeholders often ask us as ecologists to tell them is when a species is going to turn up in a new place or vanish from an old one. These are questions about the range edge, but there's a lot we don't know about range edges. And studying them also helps us understand fundamental biogeographical questions about why species are found where they are and not elsewhere, and what makes them move. I study these questions in the oceans, where species are relatively mobile, they have relatively few barriers to dispersal. We have a lot of data on these species. We have long-term, large-scale biodiversity surveys, and we know marine species are shifting faster than terrestrial ones. Before I go any further, I actually want to acknowledge that data because it's really amazing and provides the foundation for everything that I'm going to talk about today. The National Oceanic and Atmospheric Administration, or NOAA, goes out every year in the United States and goes fishing in our territorial waters, and they record everything they catch, they make that data public, and they have for decades. So everything I'm talking about today hinges on this incredible public data resource. In addition to just asking whether range edges are responding to climate and temperature change, I think it's important to ask how well they're doing it, which is something that we've done in other realms of global change biology. In this 2013 paper, using actually the same data set, the NOAA Trawl survey, Malin Pinsky and colleagues showed that there's a very strong correlation between where you would expect a species center of its distribution to have shifted based on temperature alone, which is on the x-axis, and where it actually went, which is on the y-axis. This is a really, really clear relationship, but as you can see, there's also a huge amount of variation, and species have really different responses. Some of them shifted even though they weren't expected to. Some of them shifted in the opposite direction that they were expected. Many didn't shift at all. So we're addressing these questions in 3 regions around the United States: the Northeast, the West Coast, and the Eastern Bering Sea. The Northeast and West Coast, so these dashed lines are showing the axis or metric that we use to evaluate range edge position. On the Northeast and West Coast, that was just coastal distance. The Eastern Bering Sea has a complex coastline and complex bathymetry. So we just used a straight line through what's called the middle domain, which is a shallow shelf region that managers often delineate in the center of the region. We picked these regions because they have really different warming histories. The Northeast has warmed pretty consistently over the past many decades, especially in mean annual sea surface temperature, which is shown here in orange, and in maximum annual temperature, which is the red line. More specifically, that line is a monthly maximum, so it's showing you the hottest month in summer every year. The blue line is minimum winter temperature, so it's the coldest month in winter every year. The West Coast hasn't warmed, but it does have these enormous fluctuations in temperature that are related to El Nino. And the Eastern Bering Sea has warmed, at least in maximum summer temperatures, but very episodically through these huge spikes that are correlated with years with record low sea ice in the Arctic, which I'm sure you've read and heard about. The first project we did, which I'm going to cover really briefly, was just using the raw data from the Northeast and looking at range edge positions versus mean annual sea surface temperature, the orange line in this plot. We broke down our results by range edges that were poleward, or the cold range edge, versus the equatorward range edge. And we found that in the region over 50 years of warming, poleward edges had shifted further and tracked temperature change much more closely than equatorward edges. It's worth pointing out here, and this is also true for the later analysis, that these are not the range edges of the same species, because marine species have such large ranges that we almost never get the poleward and equatorward edge in the same study region. This work is published. I'm happy to answer questions about it. But what I really want to tell you about is what we did next in extending this work to multiple regions and making a number of other methodological changes. The first change was figuring out a way to compare among these regions apples to apples, because the regions use somewhat different methods and equipment to do the surveys. So we used a model called the Vector Autoregressive Spatial Temporal Model, or VAST, developed by Jim Thorson at NOAA, which in a sentence takes the raw survey data of encounters and biomass and turns it into a biomass estimate for the whole region for that species. And that's what allows us to compare data from the West Coast to the Eastern Bering Sea and the Northeast. So I'm just going to dive in and show you what we found. So I'm not going to do that. There's one more methodological change that we made that I think is important to talk about, which is how we conceptualize the relationship between range edge position and temperature. Previously, we used mean annual sea surface temperature, but it's actually more likely that range edges would be limited by extremes, either warm or cold extremes. It's easy to assume that the cold or poleward edge is limited by cold temperature, it's in the name, and the warm equatorward edge is limited by warm temperature. But as beautifully shown by this diagram from 1947, it's theoretically possible for either one to be limited by either temperature. You can see here that yes, cold can limit poleward survival and heat can limit equatorward survival. But it's also possible at the poleward edge that summer is not warm enough for repopulation and growth. And similarly, that winter at the equatorward edge is too warm for those processes. So we related range edge position to the red and blue lines that I showed you earlier, which were maximum and minimum temperatures in the region. Now I'll show you what we found. In the Northeast, so this plot is showing you equatorward edge species in the top panel, poleward edge species in the bottom panel, and the x-axis is just edge shift, so how far every year we estimated that edges were moving. And the y-axis shows which of the elements of the edge thermal niche, warm, cold temperatures, the edge was tracking. The first thing that we found, which we were very surprised by, was the opposite result from the previous paper. Here you can see that the equatorward edge points are further to the right, which means that they shifted further. This was largely driven by these points that tracked neither temperature extreme. These are equatorward edges that retracted really far north, much further than expected based on temperature alone, into much colder water. Some of these are species that you might have heard of, like Atlantic cod and American lobster, which possibly have an interplay between a climate response and a population dynamic process. And we found some species that were tracking each thermal extreme and many that were tracking both. At the poleward edge, we also had a number of species that went south. So that's also a range retraction. It could be a population dynamic process. Definitely not climate related, because they're going in the wrong direction. We don't really know why that's happening. The results for the other regions are easier to present because basically every species was classified as tracking both temperature extremes. I would not interpret this as conclusive evidence that these range edges will perfectly adhere to these edge thermal niches through all future environmental scenarios. Rather, because temperature hasn't really changed in the West Coast during this time period, we can't rule out the possibility that the position of these range edges is associated with winter or summer temperature. And we actually found the same result in the Eastern Bering Sea. Even though summer temperatures did increase, that increase wasn't enough for most species, or they tracked it to a certain degree to push them out of this category. So they all ended up being classified as tracking both elements of the edge thermal niche. There are a few take-homes from the study that I want to share with you. The first is that this is one of the biggest studies to look at range edges that we know of. We had about 140, and it's not surprising that with more data, we had a smaller effect size. It's often observed in ecology that studies with the fewest data points often have the biggest effect sizes. But in places where we had inconsistent warming, the West Coast and the Eastern Bering Sea, we can't really rule out the possibility of tracking the edge thermal niche. Range edges may or may not be doing that, but until we can tease apart those effects better, for all we know, they are tracking temperature. Where we could tease apart those effects, the Northeast, we saw the most evidence for non-temperature processes, like with the poleward range edges that shifted south. So against that backdrop of continuous warming, we were able to identify which range edges were tracking temperature much better than others. and what element of temperature they were tracking. We didn't find that polar edges always are associated with cold temperatures and equator edges are always associated with warm temperatures. So I think it's important with both elements, both parts of the range, to study both sets of temperature extremes. We found extremely individualistic species responses. They were all over the place, but in at least the Northeast, and ideally in more regions with longer time series and more data, we can identify which species are tracking the edge thermal niche better than others. So to summarize, we developed a new method to quantify range edges across regions and relate them to temperature via this edge thermal niche concept. In doing this, we found that the region with the most consistent warming also had the least evidence for edge thermal niche tracking, which suggests that non-temperature processes might be important there. But until and unless dramatic warming reveals which range edges are tracking temperature better than others, we can't rule out edge thermal niche tracking for any of them. And finally, this question of whether poleward versus equatorward edges are shifting at similar rates in the oceans, I think is still an open one, and hopefully something that we'll keep studying in the future. I want to really thank you for your attention, and I'd love to start a dialogue about this work and anything related to it. I've listed here all of the ways that you can get in touch with me, and I'll also be following the live Q&A.